Everyone, welcome to Anima Anatomia. So today in this video, the contents which we will discuss, these are introduction to the muscle anatomy, like type of muscles, skeletal muscle, cardiac muscle, smooth muscle, and the structure of muscles like fibers, sarcomeres, and contraction mechanism. And also we will discuss here about neuromuscular junction and how the muscle receives signals. And we also included muscle of the facial expression, muscle of mastication, and neck muscles.
Hello everyone, our topic is muscle anatomy. So muscle anatomy refers to the study of muscles in human body, including their structure, function and classification. So what is muscle? Muscle is made up of thousands of small fibers that are present together to stretching and pressing in what moves your organs or the body. There are three types of muscular tissue or the muscles found in a human body, that is skeleton, cardiac and visceral or the smooth muscle. On the basis of location, the skeletal muscle is found in skeleton. Its function is about movement, heat and posture, and appearance is striated, multinucleated or eccentric, and the fibers are parallel to each other. The control is voluntary. The second is cardiac. Cardiac, as per the name, is found in the heart that pumps the blood continuously. They are striated and one central nucleus. The control is involuntary. And the last is the smooth muscles or the visceral muscles that are present in the GI tract, uterus, eye, and blood vessels. The function is about peristalsis, blood pressure, pupil size, and erects hair. There are no striations and one central nucleus, and the control is involuntary.
So now we will talk about the muscle structures. In this, we will talk about fibers, sarcomeres, and contraction mechanism of the muscles. So firstly, let's start with the muscle fibers. Muscle fibers are the fundamental contractile unit of the muscles, composed of myofibrils, which in turn contains sarcomeres, the smallest functional unit responsible for muscle contraction. Now we'll get to know about the types of muscle fibers. First is skeletal muscle fibers, which are striated, voluntary, and multinucleated. Second is cardiac muscle fibers, which are also striated, involuntary in function, and uninucleated or binucleated. Third one is the smooth muscle fibers, which are non-striated, involuntary, and uninucleated. Each muscle fiber is enclosed by a sarcolemma, that is cell membrane, and contains sarcoplasm, which is cytoplasm, which holds organelles such as mitochondria, myofibrils, and sarcoplasmic reticulum, which is denoted by SR. Second ones are the sarcomeres, the functional unit of contraction. A sarcomere is the smallest contractile unit of myofibril. Extending from Z line to Z line, it contains thick and thin filaments sliding past each other during contraction, which are known as actin as well as myosins. Structure of a sarcomere, it includes different type of lines and bands. The first one is the Z line, which is also known as the Z disc. Define the boundaries of a sarcomere where thin filaments, these are actin, are anchored. Second is A band, the darkest region representing the length of thick filament, that are myosins, which partially overlaps thin filament. The third one is the I band, the light region containing only thin filaments, these are actins, which shortens during contractions. Fourth one is H zone, the central region of the A band where only thick filaments are present. It disappears during contraction. M line, the middle of the sarcomere where thick filaments are connected. Now we will see a diagrammatic representation of the muscle fibers. So these are the sarcolemmas, this outer lining, and myofibrils are the bundle-like structure present in between them. And for example, this is I band, A band, and this one is also I band. So the distance between two Z line is known as the muscle fiber, in which there is a sarcomeres, and these are thin filaments, these are thick filaments. So in this, actin is denoted by a small dot over there, tropomyosin by a straight line, and troponin with a colon-like structure, myosin with the two heads, and titin with a small ohm-like structure. The third one is mechanism of muscle contraction. The sliding filament theory explains how actin and myosin interacts to generate contraction. So steps of muscle contractions include excitation, depolarization, calcium release, cross bridge formation, power stroke, and detachment and reset, as well as last step is relaxations. So we'll start with the excitation, in which a nerve impulse, that is action potential, from a motor neuron reaches the neuromuscular junction, releasing a neurotransmitter which is known as acetylcholine and is denoted by ACH. Second is depolarization, in which acetylcholine binds to receptor on the sarcolemma, causing sodium influx and depolarization, which spreads through T tubules to the sarcoplasmic reticulum. Third is the calcium release, in which the action potential triggers the sarcoplasmic reticulum to release calciums. Fourth is cross-bridge formation, in which calcium binds to troponin, shifting tropomyosin and exposing myosin binding sites on actin. Myosin heads attaches to actin, forming cross-bridge. The other is the power stroke, in which myosin heads pivot, pulling actin filaments towards the M line. Shortening the sarcomere, ATP is hydrolyzed to provide energy. The sixth one is the detachment and reset, in which a new ATP molecule binds to myosin, causing it to detach from actin. So myosin is recooked for the next cycle. The last step is the relaxation, in which when the nerve signal stops, calcium is pumped back into sarcoplasmic reticulum. Tropomyosin recovers actin binding sites and muscle relaxes. Now the same cycle goes on and the muscle contraction goes on in the following steps.
Now moving forward, we will discuss about neuromuscular junction and how muscle receives the signals. First of all, we should know the definition of the neuromuscular junction and its components. So the neuromuscular junction, it is a type of synapse where neuronal signals from brain or spinal cord interacts with the skeletal muscle fibers and causing them to contract. And the activation of many muscle fibers together can cause this muscle to contract, which in turn can produce movements. Now we will talk about the components. First of all, we should know what each muscle is surrounded by. So we have the three layers: epimysium, perimysium, and endomysium. So what is the epimysium? Epimysium, it is a thin sheet of connective tissue or fascia. And perimysium, within the muscle, bundles of each muscle fibers which are known as fascicles. They are bounded together by another layer of connective tissue. And then we have the endomysium, in which every muscle fiber within a fascicle is itself encased in a layer of connective tissue. In this diagram, you can see this is the bone and this is the muscle. And bone and muscles, they are connected to each other by the help of tendon. Now, the layer of this muscle, outer layer, it is called epimysium. And in that, we have the perimysium, and each fascicle consists of the muscles, fibers, and these are they have the layer called endomysium. Or you can see in this diagram also, this is the muscle fasciculus, and the outer layer, it is called the epimysium. And then these muscle fasciculus have the outer layer, which is known as the perimysium, and in that, you can see here, in that fascicles, we have the inner layer called endomysium. Now we have to talk about the other components. Other components include axon terminal and the motor end plate. We will briefly discuss this because this is mainly the topic of physiology, but we can discuss the definitions here. Now, the terminal branch of nerve fiber is called axon terminal, and when axons comes to the muscle fibers, and it loses myelin sheath, and the slender is exposed, then this portion expanded like a bulb, which is called as motor end plate. Then we have the synaptic trough or gutter. Now the motor end plate invaginates inside the muscle fiber and forms a depression known as a synaptic trough or synaptic trough. Now, then we have the synaptic cleft. Now, membrane of the nerve ending is called presynaptic membrane, and the membrane of muscle fiber is called postsynaptic membrane. And the space between these two membranes is called synaptic cleft. Don't worry, we will see this in the diagram. You can see it here. Like this is the axon, and this is the axon terminal. Now, this is the muscle. As I told you, the membrane of the axon terminal, it is called as presynaptic membrane, and the membrane of the muscle fiber is called postsynaptic membrane, and the space between these two membranes is called the synaptic cleft. Here you can see synaptic cleft, postsynaptic membrane, and presynaptic membrane. Now we will discuss about the events of neuromuscular transmission, like how the muscle receives the signals from the axon. It occurs in the 5 events. First is the release of acetylcholine, second is the action of acetylcholine, third is the development of end plate potential, then development of miniature end plate potential, and the destruction of acetylcholine. We will discuss very briefly about this topic. So here is the diagram of the neuromuscular junction. This is the axon. You can see the axon here, and this axon have the terminal, which is bulb shaped. Now we have this membrane or the layer of muscle. So what happens here? I will tell the mechanism first. Now, so what happens here, when the action potential comes from the nerve ending, from the axon, when action potential reaches the axon terminal, it opens the voltage gated calcium channels. These are the voltage gated calcium channels. So when the action potential comes, these channels are opened, and the calcium ion which is present in the extracellular fluid, ECF, they will come inside the axon terminal. You can see the calcium ions inside the axon terminal. Now what happens, these calcium ion which came from ECF, they pushes the vesicles. These are the vesicles which contains the neurotransmitter acetylcholine. So these vesicles are shifted towards a presynaptic membrane, and then when calcium pushes it to the presynaptic membrane, these vesicles ruptures by the process of exocytosis, the acetylcholine is released into the synaptic cleft or the synaptic gutter. Now these acetylcholine, so this is the first phase, that is the release of acetylcholine. Now the second is the action of acetylcholine. So these acetylcholine, after entering the synaptic cleft, acetylcholine molecules bind with the nicotinic receptors present in the postsynaptic membrane. Here you can see this is the postsynaptic membrane, and in that postsynaptic membrane we have the nicotinic acetylcholine receptors. So you can see these are the receptors, nicotinic acetylcholine receptors. So what happens? These acetylcholine binds with these nicotinic acetylcholine receptors, and due to this, the what happens? The permeability of the postsynaptic membrane increases, and sodium ions, which are present in the extracellular fluid, they comes inside the muscle by the ligand-gated sodium channel. Now, after the sodium ions alter the resting membrane potential and develops the electric potential, this electric potential is called end plate potential. Now, we have discussed the second part also. The second point, action of acetylcholine. And the other points we will discuss very briefly. I will tell you, like end plate potential is the change in resting membrane potential when an impulse reaches a neuromuscular junction. And the resting membrane potential at neuromuscular junction is minus 90 millivolt. And when sodium ions enter inside the site, the depolarization occurs and it can changes up to minus 60 millivolt, which is called end plate potential. Which is called end plate potential. If you want to read it like in detail, you can go through the physiology. Now the fourth point is the development of miniature end plate potential. So what is it? The miniature end plate potential is a weak end plate potential in neuromuscular junction that is developed by the release of a small quantity of acetylcholine from the axon terminal. And each quantum of this neurotransmitter produces a weak miniature end plate potential, and it can be up to the 0.5 millivolt only. 0.5 millivolt. Now, about the fifth point, that is the destruction of acetylcholine. So, acetylcholine released into the synaptic cleft is destroyed very quickly within the 1 millisecond by the enzymes. The enzyme which destroy the acetylcholine is the acetylcholine esterase, because acetylcholine have to be destroyed, otherwise the muscle will contract again and again and it will loses its property. So to be in the normal position, the extra acetylcholine which is present in the synaptic cleft have to be destroyed by this enzyme. Now the acetylcholine is so potent that even this short duration of 1 millisecond, it is sufficient to excite the muscle fiber. And the rapid destruction of acetylcholine has got some important functional significance, like it prevents a repeated excitation of muscle fiber and allow the muscle to relax. If you can think that if acetylcholine again and again go and binds with the nicotinic acetylcholine receptors, the muscle will excite and contract. And if the muscle will contract for 24 hours, it will be like it should need relax or rest. So these acetylcholine esterase, uh, what it does, it destroys these acetylcholine and helps the muscle to relax. So these are the 5 events or the 5 points by which the muscle receives the signal. Now you can we can simply take a chart over it. We will discuss very briefly in a like short way how what the events occurs and where they occurs. First, in the motor nerve fiber, the action potential develops. This action potential reaches the axon terminal, and in axon terminal, what is it? It does opening of the voltage gated calcium channel. Those calcium will enter the calcium ions from ECF into the axon terminal, and these calcium ion, what it does, it opens the vesicles and release of acetylcholine by the process of exocytosis. Now, in the synaptic cleft, this is only the passage for acetylcholine, from where it goes to the nicotinic acetylcholine receptors. And on the postsynaptic membrane, these acetylcholine binds with the nicotinic acetylcholine receptors and formation of acetylcholine receptor complex. Now, due to this, the permeability increase and opening of the ligand-gated sodium channel occurs, due to which the sodium from the extracellular fluid (ECF) enters the postsynaptic membrane and development of end-plate potential occurs. Now, in the muscle fiber, what happens? This generation of action potential, it causes excitation contraction coupling, due to which the muscle will contract. If you want to read this excitation contraction coupling, you can go through the book of physiology. So this is all about the neuromuscular junction and how the muscle receives signals.
Moving forward, our next topic is muscles of facial expressions. These are the subcutaneous muscles, and the name is orbicularis oculi, orbicularis oris, and zygomaticus major and minor. The origin of orbicularis oculi is the medial orbital margin and lacrimal bone. The insertion is about the skin around the orbits, particularly eyelids, and the action, it closes the eyelids, the blinking and squinting movements. The blood supply of orbicularis oculi is done by the facial artery and its palpebral branches plus the lacrimal artery, that is a branch of ophthalmic artery. The second muscle is orbicularis oris. Its origin is maxilla and mandible. The insertion is in the skin and the mucous membrane of lips, and the action is it closes and protrudes the lip. Its blood supply is done by the facial artery, particularly through the superior and inferior labial branches. And the last is zygomaticus major and minor. Its origin is zygomatic bone and the insertion is on the corners of the mouth or called as modulus. Its action, it elevates the corners of the mouth, the smiling movement. The blood supply of zygomaticus major is done by the facial artery, majorly the zygomaticus branch. And the zygomaticus minor blood supply is also done by facial artery or the buccal branches.
So now let's talk about the muscles of mastication, which includes basically three muscles, and these are masseter, temporalis, and pterygoid. And pterygoid also contains one that are lateral pterygoid and medial pterygoid. So let's start with the masseters. So masseter muscle. These are quadrilaterals, covers lateral surface of the ramus of mandible, has three layers also, and it origins from the superficial layer that is the largest from anterior two-third of lower border of zygomatic arc and adjoining zygomatic process of maxilla. Second one is the B, deep layer from deep surface of zygomatic arc. C, that is middle layer from lower borders of posterior one-third of zygomatic arc. You do not have to study about the fibers, so let's move to the insertion of masseter. First one is the superficial layer into the lower part of the lateral surface of ramus of mandible. The deep layer gets into the rest of the ramus of the mandible, and the middle layer into the central part of the ramus of the mandible. The nerve supply of masseter muscle is masseteric nerve, a branch of anterior division of mandibular nerve, and its action is to elevate mandible to close the mouth to bite. Superficial fiber cause protrusion. Next one is the temporalis, which is a fan-shaped, fills the temporal fossa, and the origin is temporal fossa, excluding zygomatic bone and temporal fascia. So no need to learn the fibers. Next one is the insertion, margins and deep surface of coronoid process and anterior border of ramus of the mandible. And the nerve supply includes two deep temporal branches from anterior division of mandibular nerve. The action of temporalis is elevates mandible, helps in slide to slide grinding movement, and posterior fibers retract the protruded mandible. The other one is the lateral pterygoid, which is a short conical has upper and lower heads. The upper head, now we'll talk about the origin. Upper head, that is small, from infratemporal surface and crest of the greater vein of sphenoid bone. The lower head, that is the larger, from lateral surface of lateral pterygoid palate and origin in medial to insertion. Now, about the innervation, or you can say the insertion, the pterygoid fovea on the anterior surface of neck of mandible and anterior margin of articular disc and capsule of the temporal mandibular joint. Insertion is posterolateral and at slightly higher level than origin. Now, the nerve supply includes a branch from anterior division of mandibular nerve, and the action of lateral pterygoid is to depress mandible to open mouth with suprahyoid muscles. It is indispensable for active opening of the mouth, and it protrudes mandible. The other action is right lateral pterygoid turns the chin to left side. Now, the last one, medial pterygoid, which is quadrilateral, has a small superficial and a large deep head. So, the origin is superficial head, that is small slip from tuberosity of maxilla and adjoining bone. The other one is the deep head, that is quite large from median surface of lateral pterygoid palate and adjoining process of the palatine bone. Now, the insertion is roughened area on the medial surface of angle and adjoining ramus of the mandible, below and behind the mandibular foramen and mylohyoid groove. The other one is the nerve supply, in which nerve to medial pterygoid branch of the main trunk of mandibular nerve. And the action is it elevates mandible, helps protrude mandible, and right medial pterygoid with the right lateral pterygoid turns the chin to left side as part of the grinding movement.
Our next topic is neck muscles. The first muscle is the sternocleidomastoid muscle. Its origin is from the manubrium of the sternum and the medial third of the clavicle. Its insertion by a thick tendon into the lateral surface of the mastoid process from its head tip to the superior border, and by a thin aponeurosis into the lateral two-third of the superior nuchal line of the occipital bone. Its action, when the muscle contracts, it turns the chin to the opposite side and tilts the head towards the shoulder of the same side. If we talk about the blood supply, the sternocleidomastoid muscle is supplied by the sternocleidomastoid branch of superior thyroid artery, that is a branch of external carotid artery. The second muscle is the trapezius muscle. Its origin is from external occipital protuberance, that is a bump on back of the head. The second from the ligamentum nuchae, and the third from the spinous process of C7 to T12 vertebrae. The insertion, it inserts on the lateral third of the clavicle on acromion process and the spine of scapula. If we talk about the action, its upper fibers elevates the scapula and helps to extend the neck. The middle fibers retract the scapula and the lower fibers depress the scapula. Overall, it helps to stabilize and move the shoulder girdle and supports the neck. If we talk about the blood supply of trapezius muscle, it is supplied by the transverse cervical artery plus the dorsal scapular artery and occipital arteries. The third muscles are the suprahyoid group or the suprahyoid muscle. It includes four muscles: digastric, stylohyoid, mylohyoid, and geniohyoid. So first, digastric. Its origin is from the digastric notch from the medial surface of base of mastoid process. Its insertion is on the digastric fossa and action it depresses the mandible. If we talk about the blood supply of digastric muscle, it is done by the its posterior part is supplied by the occipital artery, while overall the digastric muscle is supplied by the facial artery as well. The second muscle is the stylohyoid. Its origin is from the back of the styloid process near the base of the skull. Its insertion by the two slips into the junction between the greater horn and the body of the hyoid bone. Action, it elevates the hyoid bone, and its blood supply is done by the facial artery. The third muscle is the mylohyoid muscle. Its origin, whole length of the mylohyoid line of its own side on the inner aspect of the mandible from the medial to the third molar to below the mental spines. Insertion, anterior 3/4 into each other interdigitation and the posterior 1/4 anterior surface of the body of the hyoid bone. Action, it forms a mobile but a stable floor of the mouth, and its blood supply is done by the facial artery.